welcome to everyone. It's very exciting to be here, especially because I actually gave the first Big Question lecture in Centre of the Cell's Big Question lecture series, which was 13 years ago. And the other day, I looked back at the PowerPoint presentation I prepared, and I was really surprised and quite shocked at what wasn't in that, at how things have really changed in a good way since I gave the first Centre of the Cell Big Question lecture in 2011. And so I thought it was time to update that question. And I should also say at the start off that I'm very grateful to the 5 Youth Forum members who joined us a month or so ago. They were really helpful in organising this lecture, in my mind, especially as they wanted to know about new treatments for cancer. So I've tried to follow their guidance in this talk. But I'll start off by just explaining to all of you what we really understand by the word cancer. Now, sorry, I forgot one thing before I do that. Right, so the first thing to say in the question of, is there ever going to be a cure for cancer, is that cancer isn't a single disease. There are as many types of cancer as there are different cells in the human body. Here's some statistics from the Cancer Research UK website, last time they collected them. And you see, these are the commonest cancers in the UK in men and women. But even amongst them, there are quite a lot of subtypes of breast cancer, for instance. But in fact, there are many other types of cancer not so common. The other thing to know is that 50% of all cancers people will survive 10 years. A lot of those, of course, are quite common, easy to treat skin cancers, but even so, there's been an improvement in cancer survival over the last 40 years. It is doubled. But the other thing we must remember is there's a big variation in survival from cancer, from 1 to 99% curable. So when you ask the question, will there ever be a cure for cancer, the first thing you have to remember is that there are many different types of cancer. But the second thing we need to understand is what cancer is in the very beginning. Now, if I ask the majority of you, and I may be wrong, but I think I'm right because I've asked this question many, many times to many different types of people, many different age groups. If I say, what do you understand by cancer? The normal answer is cancer is when cells have gone wrong. Cancer is caused by mutations. Cancer is when cells go wrong and they spread around the body. I think most of you would agree with that. Yeah? And so the kind of process that we think of is that if you imagine those yellowy things with a brown bit in the middle are cells, and they're sitting on some kind of membrane, and then there's sort of other tissue underneath, and they're perfectly normal, just growing as much as they do, doing the jobs they should do. And then you get some changes. You see some changes in the DNA, maybe a mutation. The cells grow slightly differently, but they're not really causing any problems. And then things begin to look even stranger. But again, they stay where they are. They're proliferating more. They've got more mutations, but still, they're not causing much of a problem and it'd be very difficult to detect. But then things go really awry. And you begin to get what we call a tumour, and then you get the issue where they have grown a lot and they begin to invade into the surrounding tissue and spread. So that's the sort of common idea. Does that sort of make sense to you all? But that's only actually half the story. And in terms of the most exciting new treatments, there's another side to that. If you look at this, this is a thin slice through a human cancer. And it's been taken from a patient, taken to the lab, sectioned very thinly and stained with some dyes. And each of those blue spots is the nucleus of a single cell. So you can see that there's hundreds and hundreds and hundreds, thousands probably, of cells in the image you're seeing there. Now I'm going to cover up all the cancer cells. There's quite a few other cells there, isn't there? Probably about 50% of that cancer is not malignant cells at all. And it's really important to what I'm going to say tonight to remember that cancers are not just made of malignant cells. And a more accurate demonstration, a cartoon of what happens in cancer is shown here. And these little green cells that have been drawn here are actually immune cells. In fact, there are very many different types of immune cells. And very, very, very early on, when you have a few abnormal cells, you begin to see some of the cells of our immune system within the lesion. Now, these are the cells that normally heal wounds. These are the cells that normally fight infections. But what are they doing here? At the beginning, they may be trying to recognize and destroy the cancer cells. But as this process continues, you get more and more of these immune cells. But also, something is happening in the surrounding tissue. And in fact, there is some evidence, some scientists believe that the first thing that happens, the first thing that goes wrong, is not in the cells that go to form the cancer, but it's something in the surrounding tissue that stimulates abnormal growth, that causes mutation. Could be a chronic infection, it could be damage, tissue damage. We don't know. But this tumour microenvironment, as we call it, which is all these immune cells, is really critical, I believe, to the long-term cure of many cancers. So every cancer contains some immune cells, for instance. So here, again stained with blue and purple dyes, we have a breast cancer and a prostate cancer. Now, in the next images, we've used a special technique to stain the immune cells brown. You see how many immune cells are in those cancers? And look at this. This is the matrix. These blue stained are fibers, thick fibers. And in fact, many cancers are, if they've grown large enough, doctors can feel them. They're harder than the surrounding tissue. So in fact, cancers are not just made of malignant cells. They're made of immune cells, fibroblasts, and this fibroblasts that build the cells in your body, extracellular matrix, blood vessels, mediators, chemicals that allow the cells to communicate with each other, called things like cytokines and chemokines. You don't need to know their names. Many of them. These cells are all talking to each other. Fat cells. Lots of cancers grow near fatty tissues. In fact, they actually take the fat from the fat cells and use them to help them grow. And even now, and this is a really new thing, and actually I'll be at a conference in a couple of weeks' time where we're going to hear some talks on this, we now know that nerve fibers are threaded through cancers. We don't know what they're doing there, but they're there. So in fact, tumours are not just made of malignant cells, but they contain many other cell types that are recruited and corrupted to help the cancer grow and spread. And that, I think, is a message I'd like you to take from this talk, because it is understanding that and trying to change that tumour microenvironment that is leading to some of the really exciting cancer treatments at the moment. But let's start at the beginning. Will there ever be a cure for cancer? Oh, I've forgotten another slide. Now this is very special. This is a tumour microenvironment. This is taken from one of the patients at Barts Trust with ovarian cancer. It was taken to my lab, and Florian Laforet's, a postdoc in the lab, made a section about 1/3 of a millimetre, sliced it 1/3 of a millimetre and put it under a very special microscope, staining up some cells with dye. This is a living human tumour. The red dye stains the cancer cells, the malignant cells. The blue dye is staining that extracellular matrix. Purple are cells, immune cells called macrophages. We'll come back to them later on. And the purple cells, green are the macrophages, and the purple cells that you see moving around here, those are T cells. Those are killer cells. Those cells are able to recognize and destroy cancer cells. But are they doing that? No. They're either stuck with the macrophages, they're either moving on the matrix, some of them are having a little interaction with the cancer cells, but they have been suppressed, exhausted by the cancer. They can't recognize the cancer as foreign. So, how can we find a cure for cancer? Well, maybe what we do is, can we stop cancer happening in the beginning? I mean, that's a very easy way to cure cancer. Will we ever be able to prevent all cancer? Well, the first thing to say is that we really think that cancers, because we have billions of cells and they're always dividing and DNA is always replicating, it's very difficult to imagine how you'd be able to prevent cancer, all cancers. But we can prevent some, and some of you may even know of an example. What about vaccines? Vaccines prevent us from getting some viral and bacterial infections. There is one very good example of a vaccine that can prevent cancer. And this is the HPV vaccine. Human papillomavirus is necessary and sufficient for some cancers. And now in this country and in many parts of the developed world, all young people, boys and girls, will have HPV vaccinations. And it is expected, and in fact data are already emerging, that 90%, it will protect against 90% of cervical cancers, anal cancers, genital warts, and throat and mouth cancers. This is enormous. Cervical cancer used to be the major killer of women all over the world. Now we have a screening process that has already reduced that issue. You wouldn't see cervical cancer in that first slide I showed you. But in years to come, this HPV vaccine in the developed world, and now the challenge is to get it into low and middle income countries, this HPV vaccine should wipe out cervical cancer. Now, unfortunately, there are not many other good examples that possibly with some types of liver cancer in years to come. But there are other ways. What about lifestyle? This is from the Cancer Research UK website. It is predicted that between 35 and 40% of all cancers are a result of lifestyle. So in this thing, it says, if 10 years ago, everyone in the UK was a non-smoker, had a healthy weight, ate and drank healthily, was more active, was safe in the sun, drank no alcohol, etc., then that GP would see 40% less cancers in a year. Now we can make healthy lifestyle choices. Environmental pollution is more of a problem. We don't know the impact of that, but it's increasingly clear that it may have an impact. And I highlight a study here run by Chris Griffiths at Queen Mary, which is looking at childhood lung health in London and Luton and the impact of ULEZ. In years to come, I think we'll know more about this, both in developing in the developed world and in the lower middle income countries. But I think it's important. And obviously, as you probably know, in years gone by, environmental pollution and industrial pollution was responsible for many cancers. So we've had vaccines, lifestyle, environment. The other thing is there are a small number of people, a small proportion, who are at high risk of getting cancer because of genes they've inherited. And finding and monitoring them means that we can get to cancers before they cause any problems. And the trouble is we don't have a screen for all cancers. But you don't need to worry about those names there, but those are some cancer genes that scientists have identified. And for breast and ovarian cancer, there can be risk-reducing surgery. Remove the organ that's going to get the cancer, job really sorted. There's medical prevention. For women with high risk of breast cancer, tamoxifen and other drugs like that can really be cancer preventative. For people who have an inherited cancer syndrome called Lynch syndrome, aspirin seems to be protective against bowel cancer. And then people who have very high risk can also be screened by things like MRI and colonoscopy, which brings me to early detection. So again, if we detect cancers in those very early stages here, before they've started to grow into big tumours with the risk of spreading into the blood and lymph nodes around the body, if we can detect them here, that's it, job done. They can be removed surgically and there is no further problem. So I've already talked about high risk individuals. We have screening programs in this country and around the world for some cancers such as breast, bowel and lung in people who are heavy smokers. But the most exciting thing that's happened in the last 13 years that really was not on my radar is what we call liquid biopsies. So multiple cancers can now be detected by DNA. When a tiny, tiny cancer is growing, it's shedding off bits of DNA that are mutated DNA. And the technology, the same technology that allows us to identify criminals from tiny fragments of clothing that were taken and stored 30 years ago, the same technology, and it's advancing so rapidly. So DNA, RNA, proteins made by cancer, you know some example of this, PSA, the not brilliant, but an indicator prostate cancer test. Metabolites, things the cancer as it grows produce, and even cancer cells themselves. In people who have reasonably advanced cancer, some cancers, they even shed cancer cells into the blood that can be detected in very sensitive assays. So this has a lot of really major implications for cancer in the future. It's normally blood, but it can be urine, particularly say for bladder cancer, saliva, and even breath, because the kind of technologies that we're seeing develop now are just allowing an insight into the earliest stages of cancer without putting people in big scanners or subjecting them to X-rays. It's also particularly useful if somebody, I think this is more in the future, but if somebody comes to the doctor with symptoms that maybe are indicative that they might have cancer, a very rapid blood test will sort of channel them into the right diagnostic pathway. But also, artificial intelligence will play a big role here, because maybe a tumour DNA, it's called cell-free DNA assay, together with that patient's medical records will, really highlight. And modelling suggests that this kind of technology could reduce cancer deaths by around 20%. So I'll talk about one trial that's ongoing in the UK, and this is called the Gallery trial. And I think this is a very clever trial. They've recruited 140,000 people, and they've chosen areas where normally cancers present late, where there's more cancer deaths than other areas. And then they've recruited these people and they've randomly put them into two groups. One group is tested every year, I think for 4 years. The other group, blood is taken, but it's not tested. And at the end of 4 years and ongoing studying them, they're going to look to see if cancer deaths have been changed. These are people, I think 50 to 75, if I remember correctly. So I think you can see what will happen here. So if someone in either group gets a cancer, the hypothesis that the scientists have set is that the ones that are having this DNA test will be picked up earlier and maybe have that kind of treatment that means they can have surgery and cure. But there are studies like this and other studies, even population screening. There's a study coming from Professor Manchanda at this university in Yorkshire, where they're screening a whole cohort of women for breast cancer and ovarian cancer susceptibility genes. Just a random selection of the population, and then they can help them get prevented or early detection of cancer. I think in the end that this kind of technology is going to make a really huge impact. We can even talk about wearable devices. I don't know how often you wish to be screened for cancer, but it just, in years to come, a lot of cancers will be prevented from ever killing people because they'll be detected early. But there will come a point, certainly at the moment, when sadly a cancer is detected late, and the most difficult to treat cancers are those where we don't have a test for very early cancer, and maybe a cancer that spreads very early and very rapidly. And then we have treatments. So I've looked here, especially after the conversation with our Youth Forum members, about what's really exciting about new treatments. So if a cancer is early, it hasn't spread, or even if it has spread and you want to reduce the cancer burden, surgery is still a very important treatment. In fact, surgeons would say they cure more cancers than anyone else, and at the moment they're probably true. I'm not going to talk any more about that, but when I talk to cancer surgeons, the things that really excite them at the moment are obviously the robotics. Because one thing that's very practical that I hadn't thought about, because surgeons have to sort of go like that the whole time and they all get bad backs. But with the robotic surgery, not only can they be more precise and they can do minimally invasive surgery, patients recover quickly, and also it's good for the surgeons' backs. But there's lots of fantastic advances in robotics, particularly for cancers like prostate cancer. Now, chemotherapy is still the mainstay in many cancers, but I think that slowly that will be replaced. And as I will explain in a moment, there's ways of refining and using the chemotherapy drugs that we have in a better and cleverer way. When I gave this talk 13 years ago, I talked about targeted treatments. Now, targeted treatments are based on the oncogenes, on the cancer-causing genes that we know are mutated in cancer cells. So they target that oncogene that the cancer cell needs to grow, the cancer cell needs to spread, much more specific. And some of them are fantastic and really important and have long-lasting effects. Unfortunately, many of them, there's a problem with resistance. And it really hadn't been considered when these targeted treatments were developed. If you imagine, the analogy I once heard, which I think it makes sense to me anyway, is you imagine a bucket that's meant to hold water. It's got a hole in it, and so it doesn't hold water. You patch up that hole, and it's great, your bucket's working. But what you don't realize is that the whole of the bottom of that bucket is weak, and it begins to sprout other holes. So what happens with targeted therapy is that it wipes out the cancer all around the body, but cancer's a moving target. There are clones of cells that got different mutations, and they overcome that very specific block, and they develop other mutations, and clones with other mutations grow. So in many cancers, in fact, the metastatic melanoma, the skin cancer that spreads, it's a very deadly cancer. When I talked about cancer treatments 13 years ago, I was talking about targeted treatment for melanoma. And it was very exciting. The cancers would dissolve away, but they would come back. But my clinical colleagues told me that some of the really, even the most powerful oncogenes that were undruggable, there's some amazing new chemistries out there that are really going to make a difference in the future. And the other one that I will just talk about but won't show you anything about is radiotherapy. Again, the proton beam that you may have heard of, very focused, much more accurate, less toxic radiotherapy is making a big impact, and the technology there is also improving greatly. But what I want to talk about is much more about the treatments that are in focusing on the tumour microenvironment. And they're listed here, but I'm going to go through them one by one. But I want to start, before I show you some of these results that come from the very latest scientific papers, I want to show you what we would call is a stylized version of something called a Kaplan-Meier curve. So basically, going up the side here is the percent survival. If you imagine at this point, this is where your patients are all diagnosed, and this is the time to when they will sadly die. And this is a stylized summary of what's happened in metastatic melanoma. Melanoma has spread over the last 13 years. This is what would happen with those patients having chemotherapy. And this probably is just months. There will be lots of events down here, which is sadly patients dying. This is the impact that targeted therapy had. So you can see that the patients are living, well, that's about 50%. The 50% survival of those patients is a few months more. Then came a discovery made by scientists, particularly somebody called Jim Allison and somebody called Tasuku Honjo, and many other scientists. They discovered that when our immune system fights an infection, or when it is healing a wound, then there comes a point when you want to switch the immune system off. The immune system is extraordinarily powerful. And so the body has mechanisms called checkpoint blockade that actually stop the immune cells working, particularly those T cells, those killer T cells that I told you about before. And what Allison and Honjo and their colleagues discovered was that cancers corrupt. They take over that system, and they use immune checkpoint blockade to stop T cells recognizing and killing cancer cells. So they made very specific molecules called antibodies to that immune checkpoint blockade. They did some experiments in vitro in the lab, they did some experiments in mice, and they went into patients. And with the first immune checkpoint blockade, the first type of immunotherapy I'm going to talk about, what happened is that. So although a lot of patients died, there was this about 20% of patients who carried on surviving and carried on and carried on. And for the first time, the word cure and cancer began to be very, very carefully used in the same sentence. Then over the last 10 years or so, what clinicians have done is adding in combinations with that immunotherapy, maybe 2 different immune checkpoint blockade molecules. And now with metastatic melanoma, there is a proportion of patients who really can be said to be cured. These are patients who would have survived just for a few months before. And what's happening is that they're giving patients immunotherapy earlier and earlier and earlier. Now, this could be true of other cancers. I want to stress that chemotherapy is still an important treatment, a useful treatment for many cancers. And this time here could be months or years. And immunotherapy is only working, it's working in a whole host of different cancers, but only a proportion of patients is responding this well. And that's one of the big challenges, is to understand how to make it happen in more cancers. Now I'm going to give you some specific examples. Now, if you've understood those survival curves. The first one actually is a homegrown. This is Peter Slotterek, and I'm very fond of Peter because he actually started in my lab. He's a clinician and he did his PhD in my lab, and he's working on something else. But while he was in my lab, he discovered that some cancer cells lack an enzyme called arginine succinate synthetase, which is AS for short. And we used to tease him, you don't want to work on AS, it's a metabolic enzyme, me in particular. I said, no, there's nothing going to come of that, you know. But anyway, Peter's very, very, very persistent. And this is 20 years. He fought with lab experiments, trying to find pharmaceutical companies. And basically what he discovered is that some cancer cells can't make a vital amino acid called arginine. And so he worked with industry to find a drug that would block arginine. Now, the survival curves I'm going to show you now from a trial called Atomic Meso and a paper published this year that he presented at a big American conference and various other, may not look very dramatic. But this is in mesothelioma, which is a cancer caused by asbestos, which is one of the most difficult to treat cancers. And this is what Peter found. This is over 200 patients. And here are patients who are treated with chemotherapy, and this is their survival. And here are patients who've had chemotherapy plus arginine deprivation treatment. And again, can you see that shoulder? that shoulder of patients who are still surviving. And these are patients with mesothelioma, and they're now alive 57 months after diagnosis. There's not many of them. But what Peter was telling me the other day is that he thinks it's the genetic makeup of the patients themselves that's helping this treatment work. This is the first metabolic treatment, targeting metabolism treatment for about 30, 40 years. A long way to go, but it's a little indication of hope. And there you can see it's quadrupled the survival at 36 months, and the trial is going on. Now, the other interesting thing at the moment, to come back to chemotherapy. Chemotherapy is very, very toxic. But what is happening, and I just give you one example, but there are many, is that people are able to link the chemotherapy to an antibody that latches directly on the cancer cells. And this is a study published last year in The Lancet. And the yellow survival curve here, again, hundreds of patients, are patients having any treatment that the physician thinks is the best treatment. These are international trials happening all over the world. And here are patients who are having an antibody plus a chemotherapy drug. And look at the difference. This is progression-free survival. This is a patient surviving without any problem, and even overall survival, there is quite a big difference here. There are many other studies like that. Well, what's quite interesting here is it's even working on brain metastasis. And then this is another very much homegrown, Tom Poles from Barts Cancer Institute and the NHS Trust. And these are papers published in the New England Journal of Medicine, which is the top medical journal in the world. And Tom has really been pioneering immunotherapy. And these are two of his latest papers. And this really just shows how things are combined. So this is invasive bladder cancer that spread. Again, very, very difficult to treat. This is progression-free survival with chemotherapy-treated patients, and this is a combination of an antibody and a chemotherapy drug. And here, what he's done, so this looked interesting, again, because he's got some patients who are long-term survivors, but then what they did was they added in pembrolizumab, which is one of those immune checkpoint blockade molecules. So the combination of the two, of now he's using an antibody linked to a chemotherapy drug plus another antibody that's waking up the killer T cells. Now, you may have heard of CAR T cells, and these are really, I hear more the use of curing cancer with CAR T cells than anything else. CAR T cells only seem to work at the moment in blood cancers like lymphoma and leukemia, but they particularly are curative in children with lymphocytic leukemia who failed all other treatments. So what happens with CAR T cell therapy, it's very individualized. The patient's blood is taken and those killer T cells are extracted, but then they're genetically engineered to home in and kill that person's cancer. And it's particularly works well in this type of blood cancers because they express on their surface something called CD19. It doesn't matter, you don't have to remember that, but basically they are genetically engineered, then the killer T cells are then grown up, millions and millions, put back into the patient, and they home in and destroy the cancer cells. And this is changing. This is Professor John Gribben and Professor Jessica Okasan. sent me these slides. They are really internationally famous hemato-oncologists, and they sent me this. This is lymphoma, a type of lymphoma, blood cancer, that is in patients who've relapsed and not responding to any treatment. And this is what happens with conventional treatment. And then this is what happens in those patients treated with CAR T cells. So you see we have 50%, 53 alive at 2 years, 43% alive at 5 years. And this is absolutely unheard of. And they look at the difference between this published in 2017 and this published just last year. And things are improving the whole time, but they don't work in the many common cancers, and I'll come back to that in my last few slides. But the big new thing, and you may recognize these two people, the founders of the scientists who founded BioNTech. They stopped their work on cancer vaccines when the COVID pandemic came in, but their work is now going on full tilt. And what happens is that each patient's tumor is taken, DNA and is extracted, what mutations, they find up to 30 mutations. They make RNA, the messenger molecule from DNA. They make RNA to, say, 30 different antigens that tumor might express, neoantigens as we call them, that are not present normally in the body, put them into nanoparticles, and then they're injected into the muscle. And what happens is the muscle cells make those cancer antigens and wake up the immune system to recognize. So what happens is that, and this is happening, I think you may have read in the newspaper about a lung cancer patient who was the first to get one of these cancer vaccines. And these are not to prevent cancer happening, but these are to stop cancer coming back. And so what will happen with these trials, which are going on all over the world really, is that the cancer patient will have treatment, whatever, to remove the majority of the cancer. And then along maybe with some other treatment, they have the cancer vaccines. Apparently these are cheap and easy to make, and they're individualized. So this is the trial that started it all off a little while ago. But this is just one type of immune checkpoint blockade in melanoma. And then this is what happened when the patients were given the one immune checkpoint blockade plus the mRNA vaccine. You see the number of patients in each arm. And what was absolutely fascinating to me was, look at this line here. Now these are patients who had that treatment, but also at the end of their first treatment and throughout this treatment period, they had no circulating tumor DNA. So that ctDNA that you may remember, I told you could be used to detect cancer in the first place, can also be used to detect cancer at the end of the first cycle of treatment. And you look, these are the patients who had ctDNA and immune checkpoint blockade, and look at what's happened to them. Really bad prognosis. These they had circulating DNA and the vaccine and the immune checkpoint blockade. But then this is the really good group. So the treatment worked really well. And I think what this tells us is that the treatment has to get rid of the vast majority of the cancer. And then these vaccines may keep it away. And that's, you see, 2024. This is very recent data. So, if you remember what I showed you before, where we are now is this. So the big question is how we can get to there. I showed you little hints of how we might do. So in the last few minutes, I'm going to tell you about one tiny bit of the jigsaw, which is the work we're doing in my group. It's only everyone just adds a little tiny bit to this jigsaw, as I hope you can see, because I've talked about so many different treatments and so many different approaches. But I work on the tumour microenvironment. So my question is, will new treatments come from our better understanding of the tumour microenvironment? Because I just talked about the T cells. I talked about the lymphocytes, but there are other cells in the tumour microenvironment. And I'll draw your attention to this again, this live tumour from one of our ovarian cancer patients. And the green cells are all the macrophages. And most of them are stuck in the extracellular matrix. Some of them bunched away here, some of them up here. See how they're holding back those lymphocytes. The macrophages are generally bad in cancer. They've been corrupted, but they are capable of waking up the T cells or even killing cancer cells themselves. So about 10 years ago, with funding from the European Research Council and Cancer Research UK, we decided to deconstruct high-grade serous ovarian cancer, which is the commonest type of ovarian cancer. We have some treatments for it, for some subgroups of patients who have particular mutations, but it presents late and it is still difficult to treat. So we pulled it apart, found out lots of things, and then tried to make better model systems where we could test more readily immunotherapies. And one thing that we did was begin to build mini tumours. Now this cancer grows particularly in a fatty tissue called the omentum in our peritoneums, and so we made cultures of malignant cells, fibroblasts and fat cells, and they made lots of extracellular matrix. Then we made 4-cell cultures where we added lining cells called mesothelial cells to those, and then Bayer Malacreda in the lab at the moment is building pentacultures, 5-cell cultures, which also have those macrophages, and this is what they look like. This little slices through them, and they do resemble the little tiny tumours that seed the omentum. But something rather exciting happened just recently. We did the same technique on them as we did on our live human tumours that we get from the patients. And I only saw this today, but I had to show you because it is so exciting. Look at these macrophages moving around. Look at this one here. So the red again are the tumour cells, the blue is the extracellular matrix, green stains the fibroblasts and the mesothelial cells. We haven't got lymphocytes in there, we haven't got that sophisticated yet. But just look, it is so exciting because we can really, I think, begin to probe and treat with drugs. I never thought we'd see this in our mini tumours like we see in the live patient ones. And then here's another one. I'll only show you two, but I am rather excited about them. I could put them on my television screen at home and just watch them for hours. Look at this one here. Can you see that one? That macrophage is really on a mission. So here, this is a different cells from a different patient, the malignant cells. These are, and they're red again. We've got this matrix, we've got the fibroblasts and the mesothelial cells, and these macrophages are moving around. The other one, the macrophages are beginning to kill the cancer cell. In this one, they're not. So we're just so excited by this. But seriously, what we're doing here is that we want to try and understand how CAR T cells, why CAR T cells are not working in what we call the solid cancers. They don't work in ovarian cancer. So our small contribution to this is here. You see the size of that's a vial about that big. You can see how tiny the mini tumours are. We put them in what we call this chip. You see how tiny it is, and they go there. And then what Joash Joy does is he puts blood vessel cells in the middle. He puts the mini tumour in this little hole here, and the blood vessel cells grow into proper blood vessels, and then he adds CAR T cells in, and the CAR T cells go in to the tumour, the mini tumour. And we found 2 reasons why the CAR T cells aren't killing some of the mini tumours. One is some of the cancer cells are mutated, so they can't be killed. They've got anti-death mechanisms by the CAR T cells. But also some of the cancer cells are making so much matrix that the CAR T cells get stuck outside the mini tumours. So we're using our mini tumours to understand what stops T cells killing cancer cells in human tumours. There's lots of mouse models that look at CAR T cells, but you have to use human CAR T cells in the patients, and so it's very exciting. And also a lot of the treatments, the biological therapies, the immunological therapies are human antibodies, so we have to have human cells. It's only one type of model, but we hope they will be useful, and it will allow us to test combination treatments faster, I hope. And just finally, I talked about the extracellular matrix, and in our mouse tumours, this is a technique called cell dive that carried out here at the Blizzard, and Bella Kotantaki, Florian Laforetz, and Eleni, who's in the audience, have been working on our tumours to see what happens if we dissolve the matrix. Now, all the colours you can see in this image are all different components of the tumour microenvironment, and we need a computer to really understand. But I think you can see without any computer that when we add a treatment to mice with these established ovarian cancers that targets the matrix, we completely change. All the yellow here's the fibroblast and the matrix, and we completely eradicate it. That's so exciting. Unfortunately, it doesn't do anything to the survival of the mouse. It brings in loads more immune cells. They can get in there, but they're still knackered, they're still suppressed, they're still corrupted. But maybe combining that matrix targeting treatment with an immunotherapy will have that kind of effect on the shoulder of the curves. So there's lots of known unknowns, and these may be questions that have arisen to you. These are questions that I've asked my colleagues. There are many more things to discover, but I think for me, looking back to my lecture 13 years ago, it really is a hopeful time. And so what we can dream of is that. I don't think personally it's going to be possible for all cancers, but you don't just have to trust me, because I got advice from all these people who work here at Queen Mary, University of London, and who are cancer specialists. And I asked them, will there ever be a cure for cancer? And these are their answers. And what is nice is they are all positive. And the majority of those people see cancer patients every day. So I think that's quite exciting. I'd also like to thank Reza, who did those diagrams for me, and also my own group and all my funding. So thank you for listening. I'm really happy to answer any questions either now or I'll be outside later. Thank you.